Today's paper. By Henjong Han. Today's paper is, Paediatric Genomics, Diagnosing Rare Disease in Children. Published in 2018. This article reviews the application of genomics to pediatric rare disease diagnosis. There are 4 topics in this review paper. 1. From genetic tests to genomic tests. 2. Integrating and interpreting data. 3. Considerations for clinical application. 4. Future perspectives. As this article is too long to cover in a short video, I will only cover 2 out of 4 topics in this video. Before we begin, there's something you should know. A rare disease is any disease that affects a small percentage of the population. There are about 7,000 rare diseases that we discovered. There is no single definition of the term a small percentage, but it generally means fewer than 1 in 2,000 people. Among the rare diseases, 80% of them are thought to have a genetic cause. The majority of the rare disease is early onset, affecting children. Rare disease in childhood can be very deleterious, and 1/3 of children with rare disease die before the age of 5. Therefore, the accurate diagnosis for the patients is important. There are many benefits of accurate diagnosis, such as, 1. It can provide access to information about disease care and treatment. 2. It enables access to disease-specific support groups. 3. It enables accurate family determination for the future. Okay, let's learn about the genomic diagnosis of pediatric rare disease. The first topic. From genetic tests to genomic tests. Okay. Let's learn about how to do genetic tests. There are traditional methods that we have done in the past. If we have a candidate gene for a test, then we can do Sanger sequencing or genotyping. We can also perform karyotyping, in which we check the number and the structure of chromosomes. It is also possible to use microarray, which can detect previously discovered disease-causing copy number variations. These methods are useful, but they have some limitations. 1. You should select gene correctly before the Sanger sequencing and genotyping. 2. Small and subtle abnormalities in chromosomes cannot be found with karyotyping. 3. The sensitivity of detecting disease-causing gene using genomic microarray is very low. Now, the modern genetic testings have been revolutionized with the well-known technology, called next-generation sequencing. This technology allows massively parallel sequencing, which means reading a huge amount of sequences very fast. So we can read the sequence entire exome or even the whole genome with NGS technology. There are benefits of using NGS for genetic testing. In the case of diseases that are caused by multiple genes, it is possible to identify sequences of the genes simultaneously. NGS is considered as gold standard technology, as it can detect SNVs and indels with extremely high sensitivity. Also, NGS can be useful for detecting CNVs and other structural variants. This figure shows the characteristics of traditional and modern genetic testing methods. The modern genetic testings using whole exome sequencing and whole genome sequencing have high resolution and coverage. However, they also have some limitations. First, about 70% of genes in the genome still have no known function and hard to interpret. Second, NGS technologies are poor to detect small-size copy number variations. Third, post-zygotic mosaic variants are often missed in low-coverage data. Next, diagnostic tests based on next-generation sequencing. There are many ways to apply NGS in clinical diagnosis. The first one is targeted tests, which focus on a single gene, or multiple disease-associated genes. Targeted approaches can be clinically attractive, because it is cheap and reduces off-target findings. However, it is also possible to miss actual causal genes. The other one is a non-targeted test. This is based on whole exome sequencing and whole genome sequencing. It sounds better to read all of your genomes at once, but as more data are generated, there occur more logistical and ethical issues. Also, it is more expensive. Until now, it is not clear which one is better for the accurate diagnosis. Maybe in the future, people would come up with much cheaper methods with high sensitivity but without the ethical issues. Trio-based diagnosis. It is recommended to test family members too, especially the parents of patients. Using this method, de novo variants that only present in the child can be easily identified. So, irrelevant candidate variants can be filtered by utilizing data from parents. In this way, we can accurately find disease-associated variants. With the trio-based genetic test, the diagnostic rate can increase to over 50%. Next topic. Integrating and interpreting data. The first step of genomic diagnosis is finding variants from the patient, called variant calling step. This step is not 100% reproducible, as it involves complex processes. The result depends on the quality of test data and reference data. It also depends on the variant calling algorithm you used. The next step is annotating each variant, and this is a critical step for clinical interpretation. However, the results can also vary substantially. This is because the result depends on the software used, providers, and versions. Next, variant filtering. It is known that there are about 20,000 variants in each individual. Variant filtering is the step to find a small number of candidate variants among them. People developed many ways to do this. Using a trio approach is a common step, but you should be careful not to find the wrong inheritance pattern. Prediction of functional effects of variants can be helpful, but these predictions may not reflect the real biology. Selecting alternative transcripts can also result in a different diagnosis. Large-scale population resources, such as XAC and GNOMAD, can be used to filter common variants. However, the threshold and ethnicity of the resource can affect the result. Assigning pathogenicity to variants is a crucial but challenging step to interpret candidate variants. Using gene-disease association is a common step, but if the association is wrong, it can result in different diagnosis. You can use databases of pathogenic variations like human gene mutation databases, but it was reported that HGMD contains numerous errors. Large population datasets, such as GNOMAD and ClinVar, can be useful resources of population genome data. Databases for clinical features, such as human phenotype ontology, is also useful for categorical approaches. Following the ACMG-AMP guidelines, published in 2015, is highly recommended for establishing a framework. Platforms like Decipher can help you to capture standardized phenotypes by clinicians. Next, data sharing. It is important to share genomic data and clinical features of patients around the world. However, it is challenging because of the data volume and ethical issues. Resources like Decipher and Phenome Central facilitates data sharing and minimizes the number of variants shared per individual. Also, initiatives like GA4GH increase the scope of search. ACMG recently issued a position statement for data sharing, saying that it is crucial for improving genetic healthcare. Okay, to sum up. From genetic tests to genomic tests. 1. Modern genetic tests apply NGS technologies to improve sensitivity and the amount of data. 2. There are targeted approaches and non-targeted approaches for genomic tests. 3. Trio-based analysis is a useful approach for better diagnosis. Next, integrating and interpreting data. 1. Variant calling and annotation are crucial steps, but the results depend on multiple features. 2. Variant filtering step can provide candidate variants using many resources. 3. Pathogenicity of variants can be assigned through multiple processes. 4. Data sharing is challenging because of the data volume and ethical issues. This is the first half of this paper. In the upcoming video, let's go over the remaining part. Thanks for watching this video. Please like and subscribe and click the bell icon to get new video updates. See you next time. Bye.